Tuberculosis, or TB, is a bacterial disease caused by the Mycobacterium tuberculosis bacteria. As it can be transmitted through airborne particles such as coughing or sneezing, TB is categorized as a communicable disease. Latent tuberculosis is when a person has the tuberculosis mycobacteria in their body, but it does not make them ill or progress to TB disease. On the other hand, active TB is when a person who is infected experiences symptoms and is therefore more contagious than dormant TB. Some of the typical indications of tuberculosis includes persistent cough lasting for 2 weeks, fever, weight loss, and chest pain. This disease mainly affects those with weakened immune systems or older individuals. TB is a challenging disease to treat, especially with the emergence of drug-resistant strains of TB. That scientists and researchers around the world are working on developing new treatments using genetic engineering. One of the main challenges in controlling TB is the development of drug-resistant strains of the bacterium. However, researchers have been working on the new approaches to combat TB, including the use of a vaccine called Bacillus calmette-guerin or BCG, which provides some protection against TB, but it is not fully effective yet. Researchers have also been looking at the use of genetic engineering to create animals that are more resistant to tuberculosis in a study that was published in the journal Genome Biology. Using CRISPR-Cas9 gene editing, scientists from the Faculty of Veterinary Medicine at Northwest A&F University in China inserted the NRAMP1 TB-resistant gene into the cow genome. Importantly, the researchers' procedure has no discernible off-target impacts on the genetics of the cow, allowing them to generate live cows with improved TB resistance.
Finding the ideal location in the genome to introduce a new gene can be difficult. According to Dr. Yang Zhang, the study's principal author, the optimal area for the insertion of the gene was nevertheless carefully and methodically chosen by the researchers, with no off-target impacts on the bovine genome. Bovine fetal fibroblasts, which were employed as donor cells in a procedure known as somatic cell nuclear transfer to produce transgenic cows, which had the NRAMP1 gene added into their genomes by the researchers. When the Mycobacterium bovis, the bacterium that causes bovine TB, was introduced to the transgenic cows, the researchers discovered that the animals had greater resistance to the disease. This discovery might significantly impact animal production in regions where TB is a major problem, as well as for human health, since Mycobacterium bovis can also cause TB in humans. According to a different study, the Mycobacterium TB bacteria's genetic composition is intricate and highly developed. This study revealed that certain CRISPR-Cas proteins released by the bacteria can also serve as virulence factors, inducing host immunological responses, in addition to their well-known defensive mechanism against invading DNA. According to the study, the Cas6 protein interacts with human cells in a way that causes the activation of specific genes and pathways linked to immune responses to TB. This release of IFN from cells of persons with active TB was caused by the Cas6 protein. The bacteria is less pathogenic in mice when Cas6 is eliminated. These findings imply that CRISPR-Cas proteins play novel extracellular activities and may be crucial therapeutic development targets.
In a nutshell, CRISPR-Cas9 technology has proven to be a valuable tool in the fight against TB. While much work remains to be done, these studies offer hope for the development of more effective treatments and prevention strategies. If you think you may have been exposed to TB or are experiencing symptoms, it is important to see a healthcare provider right away. Early diagnosis and treatment can help prevent the spread of TB and save more lives. Let's raise awareness about TB and work together to fight this deadly disease. Together, we can make a difference and start a change right away.